I first met Heather, a few years ago, and she had no idea that I met her because I met her on YouTube. Actually, it was on a TED Talk that she gave on slime molds, and I was thoroughly entranced. And I actually watched it more than once, which is pretty unusual for me. And then I had an opportunity to invite her to come to our species conference, this one, and she said yes. And so she flew from the UK on this special occasion to introduce us to one of the more remarkable creatures on planet Earth. Of course, they're all remarkable, and they're all amazing, as we've been learning. But let's have a conversation with slime molds.
Thank you very much, Adam, for the introduction and for the invitation. Really, really happy to be here. It's been fascinating the last day and a half listening to the different voices of nature across species and scales. And I'd like to share a particular species with you today. The organism that I'm going to be talking about is a non-human intelligent life form. It has no voice, literally, as it makes no sound. Legally, there's no laws protecting its existence, and there's no legal, ethical requirement to work with it. And also, I said, it has no voice symbolically, as very few people know about it. It's left to its own devices, creeping around on the forest floor, seeking dark and damp corners. So the organism in question is the many-headed slime mold, Physarum polycephalum. Physarum polycephalum is one of over 700 known species of slime mold. It is a single-celled organism, so comprised of thousands, sometimes millions of individual nuclei, all shuttling around in a cellular goop, all parts operating as one single entity. It is not plant, nor animal, not even fungi. It belongs to the kingdom of the protists. And in its natural habitat, you can see it this time of year is pretty good, living in temperate woodland, feasting on rotting vegetation. So next time you're going walking through the woods, turn a rotting log or kick up some leaves that are kind of rotting on the floor, and you might find some creeping slime molds. It's a nomadic amoeba, so highly exploratory, curious organism, and impossible to domesticate. Here it is escaping from one petri dish into another, looking for new adventures. Here it is in action. Hopefully you can see with the reflection on the slides. So, having fed on a big pile of porridge oats, which is the domesticated slime mold's favourite food, it goes exploring in two directions simultaneously. When it meets itself, it pauses, recognises that it's already there, changes direction, and retreats, puts its energy into other directions to seek new territories elsewhere. So this organism has no sensory organs, it has no central nervous system, it doesn't have a single neuron to its name, but it is attributed with all manner of computational and problem-solving capabilities. For example, it can find the shortest and the most efficient route through a maze. So this is between connecting two food sources. So you can see from the left, the maze goes from being full of slime mold. When they introduce food sources at 2 points, the slime mold rationalizes its growth and forms a tubular network between the two sources. There are 4 possible routes through the maze, and the slime mold chose the shortest and most efficient. Those that were here yesterday got a little sneak preview of this one. David stole my punchline. It can establish highly efficient networks. So here it's replicating the Tokyo transport system to a degree of accuracy of around 97%, I think, is about the accuracy. So what's happening is the slime mold is growing from the big blob in the middle, which is Tokyo. Oats have been placed at provincial rail stations around the Tokyo peninsula. As the slime mold grows out, it locks onto the food sources, and over time finds a kind of an optimized network between these sources. They often called the traveling salesman problem. If you overlay the actual transport map of that region, it is almost 100% accurate. So the slime mold has replicated this system. And since this experiment, if you want to spend some time on YouTube, an afternoon or a few days, type in slime mold mapping, and it's mapped all sorts of transport routes, other kind of networks, drug trafficking routes, migration routes, evacuation routes, all manner of mapping things. So as a model organism, the slime mold has inspired, intrigued and confounded researchers from across a very wide range of disciplines. So from molecular biology and biophysics and biochemistry into computer science and microelectronics and robotics as well. I've got a couple of examples here. So on the left, we have a digital simulation of the Tokyo experiment. So an algorithm that is mapped from biological into digital form. And on the right, we have a robot that is controlled by a slime mold. So there is a slime mold in the transistor which is responding to light, it doesn't like light, and its growth behavior will affect the direction of the robot. So the world over, researchers are decoding its biological behaviours and applying these computational rules to different fields of inquiry. And it's not just scientists. Philosophers, pedagogues, management theorists are also interested in this self-organising system. And we heard about self-organisation from Simon Garnier of the Swarm Lab yesterday. So think about some of the examples that he was giving and apply them to this non-neuronal organism. So in this self-organising system, decision-making, knowledge distribution, control mechanisms are entirely decentralised. There is no one part in control. It is a non-hierarchical organism. On a recent online search, Google Scholar cited over 59,000 published academic papers on slime mold. So there are very many human heads looking at this many-headed organism, looking to better understand how it can do what it does from such simple components. And within the fields of art and design, architects, urban designers, visual artists like me, programmers, choreographers, musicians are working with the slime mold as material, model and metaphor. I don't know if David Rothenberg has ever duetted with the slime mold. Is he here? Maybe if we have time, I might challenge him to do so. Or invite, should I say. For many researchers, the slime mold begs poignant questions, exploring how it knows its world and how it operates within it. As well as being attributed with a primitive form of intelligence, the slime mold also has capacity to learn and to remember. It has a spatial memory. So through an extended body of extracellular material, as it navigates its environment, it leaves a trace behind it, indicating where it's been, a form of stigmergic mapping, similar to slugs and snails. It also has a temporal memory. One research paper tells when the researchers exposed the slime mold to cold, dry air, at regular intervals. The slime mold slowed down when it felt the change in the atmospheric temperature and humidity to conserve energy. It didn't like the cold, dry air. After a pattern was established in response to these regular interval changes, at a subsequent interval, the researchers didn't change the temperature and the humidity, yet the slime mold slowed down in anticipation of this event that it didn't like. It wanted to conserve energy for this unfavourable climate that it was expecting. And when they reintroduced the stimulus, here on the graph you can see the repeat pattern of the change in humidity and temperature. Then it doesn't change, but the slime mold slows down in anticipation. Then it goes a little bit haywire, gets a bit confused, levels out. When they introduce it again, sometime later, it triggers the same anticipatory response. More recent research takes this idea of learning and learned behaviour further, and I'll explain this experiment to you. So here the slime mold is tempted across a bridge by the promise of food. The only problem is the bridge contains quinine, which the slime mold doesn't like. So when it first encounters this repellent on day one on the right-hand side, it approaches with caution. It takes a very narrow path across the bridge to the food. On the second day, when presented with the same problem, the slime mould is a little bit more courageous and sends a wider plasmodial search party across the bridge. And so on throughout the week. You can see the slime mould becoming bolder as it becomes more accustomed to crossing this bridge with the repellent in it. So the results from this experiment suggest that the slime mould, having crossed the bridge unhappy but essentially unharmed, retained that information and applied it on subsequent encounters, a form of habituated learning. And when we compare the experimental results at the bottom with the control experiment at the top, you can see that a neutral bridge that doesn't have the repellent in it will be crossed quite confidently by a wide growth behaviour. On subsequent days, until on day 6, they introduced the quinine bridge to the control, and it sent this very narrow, tentative search party across to find the food. So it can become habituated to its environment. The researchers from this study wanted to take it a step further and wanted to know what would happen if they merged habituated slime moulds with non-habituated slime moulds. Would the slime mould transfer this environmental information to the other body, the other slime mould? So on the left-hand side, you can see 3 blobs of slime mould. In the middle, you have a slime mould blob that hasn't been habituated. It's never encountered quinine before. And it's flanked on either side by slime mould that has. Placed in close proximity, genetically similar slime moulds will fuse and form one large cell, and we can see that happening in the video. So 2 becomes 1, but when we think about this as actually millions of nuclei, it's many becomes more many. So after about 3 hours of proximity, being in proximity of fusion happening, the habituated information had been distributed between the cellular matter, and the newly assimilated slime mould in the middle demonstrated the habituated behaviour, confidently crossing that bridge. So the environmental information had essentially transferred between organisms through fusion. So the question is, how can this brainless blob do all these things? And for this, we'll take a closer look into its internal mechanisms. And this is a microscope video that I shot. It's 400 times magnification and sped up 20 times. So within the slime mould cell, as it explores its environment, the slime mould pulses in a slow rhythmic flow. A streaming motion, shuttling back and forth within this mass cell. It's moving nutrients and cell matter, first in one direction and then back in another. Each pulse lasting about a minute or 2, so yeah, I've sped it up here. This process is called protoplasmic streaming, and we saw some examples yesterday of protoplasmic streaming in other organisms. And so whilst no one knows exactly what it's doing, it is within this continuous synchronous oscillation that researchers believe the slime mould intelligence lies. This transmission of chemical signals communicating vital information about available resources, about environmental conditions, across the whole cell simultaneously. And within this unicellular structure, all parts of the organism work together to maximise resources and opportunity. There's no top-down control, only local interactions and a constant feedback loop of communication. So the slime mould intelligence is an emergent property of these interactions. And Simone yesterday told me about some research that they're doing in the Swarm Lab about the proteins that are controlling this mechanism. So it's actin and myosin, the proteins that we have in our muscles that allow us to contract. That's essentially the protein element that's driving the slime mould protoplasmic streaming. So I'm an artist, and I've been working with this organism for almost 10 years now, as a material, as a model, and as a metaphor. And over this time, I've learned from the organism through empirical observation, experimentation, and studies. And I've grown to understand its motivations and behaviours, how it responds to my cues and my interventions. I can predict its patterns of growth, and I know its likes and dislikes, but ultimately I cannot control the outcome. It is a negotiated co-creation between two very different forms of intelligent life. Artistic processes may vary, and the outcomes. I work with print, so capturing significant moments of growth. This is the point of recognition that I showed you in motion earlier. And I do a lot of time-lapse studies, observing and influencing its behaviours along the way. Time-lapse allows the organism's behaviours to reveal themselves. There's a top speed of about 1 centimetre an hour. This is fast in some biological terms, but it's not live action. You're not going to sit and watch something growing at 1 centimetre an hour. So time-lapse photography really captures what it can do. This is my homage to the maze experiment. I built a large replica of the maze in three-dimensional form and invited the slime mold to roam freely, making decisions on which turn it would take in search of food. The film shows several days of growth captured through time-lapse. And when this is exhibited, I'll show the film. Often I will show the maze as well. It's kind of about the size of a laptop. And it was filled with agar, a nice damp environment for the slime mold to grow in. And visitors were invited to feed the slime mold and then wait patiently for it to grow and find their oats. The time-lapse studies are dotted throughout this presentation, and there are many more to be found on my website and on YouTube if you're interested in seeing more. And I also work computationally with the slime mold, so I'm interested in the relationship between the biological and the technological, and how increasingly we are looking to biological models for our technological needs. And this is an interactive piece in an exhibition where gallery visitors are transformed into virtual food nodes for the slime mold to network between. So this shuttling group of people are trying to work out where they are on the screen. So through motion capture, we become virtual oats, and the slime mold networks between us. A kind of virtual hand-holding, the slime mold allowing us to connect digitally. Incidentally, the two people that I'm shuffling around with here are filmmakers, Jasper Sharp and Tim Graben, two British filmmakers who made an award-winning feature-length documentary on plasmodial slime molds and those who work with them, called The Creeping Garden. And it is a very quirky documentary about slime molds. Recommended viewing if you'd like to know more. Covers robotics, biochemistry, a musical duet between a human, a grand piano, and a slime mold, and lots of other interesting things. I'm a teacher as well as an artist, and I am very keen to share my enthusiasm for the art and science of slime mold with others. So I run a range of participatory workshops with people of different ages, backgrounds, interests, encouraging others to ask their own questions of this curious organism. Give you just a couple of examples. This boy on the right, who is wearing a London Underground tube map. He obviously gets the whole transport thing. In his experiment, he was pondering the future of slime mold research and thinking about synthetic biology. And thinking, well, if I genetically engineered the slime mold to be bioluminescent, so it emitted light, would it have a huge existential crisis because it doesn't like light? It's photophobic, yet it would be emitting light. And this was seriously pondering. So he's going to be a great synthetic biologist of the future, I think. And I love the picture on the left, a woman looking down a microscope at protoplasmic streaming for the first time. The joy of that observation. Workshop participants are encouraged to post results of their experimentation on Slimoco, which is the slime mold collective. This is an online network connecting people working with or interested in the organism. I set this up quite early on in my own experimentation as a way to connect with others. I wanted to see who else was working with slime mold, in what way, and this was a way to seek people across disciplinary boundaries and beyond academic boundaries as well. So it now comprises over 1,000 members, from people who've dedicated their life's work studying this organism to high school kids who've got a biology homework and don't know where to start. People swap experiments and cultures and building a collective knowledge in a distributed, self-organising way, of course, and that would be appropriate. And public participation is also extended to a form of embodied collective experiment. I'm thinking about how do you engage people with the wonders of these natural phenomena. So there's kind of visual and aesthetic, there's interactive, but there's also an embodied. I think, how do we get people to embody what it is like to be part of a slime mould? So the experiment, which is called Being Slime Mould, evolved as a way of engaging just some simple rules of slime mould behaviour relating to motivation, navigation, decision making. So taking some simple rules from what slime mould is doing, and then giving them to a group of humans, and then standing back and seeing what happens. So people are given a few instructions, and then we see what emerges from that. It's usually quite chaotic. People have to operate silently because the slime mould has no voice. They have to kind of improvise some kind of oscillation communication mechanism to try and then reintroduce attractants or repellents into the environment, and they have to navigate around those. Clearly, being or becoming slime mould is an impossibility for human beings. But the point of the exercise here is in the trying. A playful attempt to let go of human habits, not habitats. Well, maybe both. But a way to just let go of being human for a short while and test other ways of being and knowing as single and collective living entities. So how we operate as individuals within groups in relation to the environment. So it's an invitation to shift perspective, to take a slime mould-centric position for a short while, and to explore how communication and cooperation could be achieved by taking a slightly different approach. So by trying to be slime mould, really humans, it becomes a mirror that reflects back on the kind of essences of humanness and where we might be failing. So I see the slime mould as something that we can learn from, not by falling into anthropomorphic patterns of projecting our own sensibilities onto it, but as a vehicle through which we can examine our own behaviours and capacities within the shared ecosystem that we habit. This is an organism that is so different to us morphologically, sensorially, cognitively, yet it does display some attributes and traits that we recognise in ourselves: learning, memory, recognition. So I think it is a really valuable tool for interdisciplinary inquiry. And I'm really pleased that I'm not the only one who thinks this way. Up the road, in Hampshire, in Amherst, so Hampshire College, you may know, is known for its progressive student-centred educational framework. And there is an ongoing human slime mould collaboration which is connecting biology, art and philosophy. I'm going to quote from a published circular from the Plasmodium Consortium. In the spring of 2017, a population of Physarum polycephalum was invited to Hampshire College, appointed to the faculty position of visiting non-human scholar, and enlisted to analyse some of the world's most vexing problems. Aided by a team of student research assistants, some of whom we have here today. Big shout out for the Hampshire posse. Aided by a team of student research assistants, the Plasmodium Consortium has studied the implications of regulating borders, drugs and the environment, and has explored new approaches to eradicating food deserts and improving public transportation. Although none of the results are definitive, preliminary policy recommendations are provided in the form of letters dispatched to government officials. The letters, sent in February of this year, outline the problems and share preliminary results from the slime mould explorations. The organism, they state, is well placed to comment on human politics, as it is unencumbered by bias and takes an entirely non-partisan position. It's also able to reach decisions through collective consensus of its many nuclear parts, which clearly is something as humans have been struggling with in recent collective voting decisions in different countries. So one letter to the Attorney General, Jeff Sessions, as was, shares some research where addiction has been modelled. So here, if I explain the experiment, the research explores the politically sensitive issue of drug classification and criminalisation, and it questions the status of marijuana, seen by some as a gateway drug to harder substances, by others as a medicinal gateway to well-being. Offering the slime mould a range of options containing highly addictive substances and those with nutritional value to varying degrees, the slime mould opted for a balanced diet, choosing a middle-range combination rather than a really highly addictive substance. So it went for a medium-high, but a good dinner as well. So the interpretation of these early preliminary results is that the slime mould suggests that cannabis should be decriminalised for medical use. Another letter to the Secretary of Homeland Security, Kirstjen Nielsen, I think she is still in post, yeah, raised the question of border control rationale. So given the slime mould options between a bordered and a borderless territories, it found the closed territory non-conducive to the slime mould nomadic tendencies. I quote, "After several days, the unconstrained slime moulds were found to join together and thrive in the open border zone, suggesting that borders may be especially vital regions if allowed to develop without government interference." The suggestion to policymakers was to introduce a strip of parkland at the US-Mexican border rather than a wall. So the policymakers that these letters were sent to have not yet responded, but they've only had since February. You know, give them time. They will, I'm sure, take on slime mould perspective. So it's uncertain whether slime mould experimentation results by the Plasmodium Consortium are going to have any impact on policy. But we can hope. And you are this afternoon invited to create your own experimental models with the Hampshire Plasmodium Consortium staff and students and myself. And we're going to do a workshop where you can model a situation of your choosing, and you can also take home a new pet. So do come along. I think it's 2:30. We're going to do that. So other studies looked at questions of resource distribution, transport networks, environmental sustainability. You can find out more on the Hampshire website, each presenting an experimental setup and the policy outcome as suggested by the slime mould. And of course, the slime mould cannot directly answer our problems, our societal and our environmental issues. It's operating with its own subjective biochemical reality. It has no understanding nor cares for our human-scale dilemmas. And whilst these approaches are not to be taken too literally, clearly there is a tongue-in-cheek tone here, I think. The study has raised really important questions for the students and for the faculty, and through the slime mould, it's engaged a very diverse range of staff and students from Hampshire on multiple levels. So they're bringing together these different disciplinary ways of thinking, combining an interconnected ecological, scientific and political inquiry. So by setting up the subjectivities of a brainless superorganism in comparison with our slightly dysfunctional superorganism tendencies, the experimental project has encouraged students to explore real-world problems in really critical and creative ways. And the Plasmodium Consortium is a thought experiment in action, bringing different disciplinary tools and languages together to interrogate multi-species entanglements. And I end with this depiction of multi-species entanglements. The Garden of Earthly Delights, how many of you know this painting? lots of you. So, painted by Hieronymus Bosch 500 odd years ago, it depicts the many intertwinings of natural history and the human condition in all its complex ways. In amongst all this activity, there are apparently, I haven't spotted them all myself, but there are 22 species of slime mould that have been identified in this painting. So Bosch understood the power of the slime mould too. And at the time this painting was being made, scholarly inquiry was held under the umbrella of natural philosophy, where disciplinary distinctions did not exist in the way that they do now. As the academies of the arts and sciences have evolved largely over the last 200 years, modes of inquiry have become increasingly specialised and dislocated from each other and from wider ecological and social contexts. And I'm a firm interdisciplinarian. I'm an artist who engages deeply with scientific endeavour, and I'm a teacher who believes that we learn best through embodied and aesthetic experience. So I think we really need to bring these different methods and mindsets together to tackle our problems today. And perhaps this little alien yet relatable organism can help us reconnect with the complexity of the natural world and can assist us in shaping better eco-political futures. So let's see it for the slime mould. Thank you.